Today I will talk to you about how to rewind the biological clock. I will talk to you about epigenetics, about the nature of aging and rejuvenation. It all starts with the DNA molecule. The DNA encodes the genetic information of our species and our individual genetic information. But what methylation clocks have taught us is that the DNA molecule is also one gigantic clock in your cells. DNA methylation allows you to measure time from cradle to grave based on epigenetic modifications of the DNA known as methylation, so-called cytosine methylation. The DNA molecule encodes 4 letters, A, C, T, G, and the letter C can be modified by methylation. Here I show you a blue curve that visualizes how methylation changes at specific locations on the DNA molecule. And what you observe early in life during development, methylation rises quickly, and then later on, after age 20, it levels off to a constant rate of change. But the most striking thing is that there's this continuous readout that connects early developmental processes to old age, one biomarker. And people have used epigenetic clocks to study development, in particular how developmental processes may impact aging later in life. But researchers have used epigenetic clocks to study aging, in particular study centenarians. Now, you may have heard of so-called blue zones. These are regions on the globe where people appear to live exceptionally long lives. However, some researchers have raised doubts about it. They feel these blue zones may arise because of false age claims in order to secure insurance benefits. Well, now we have epigenetic clocks for centenarians. We can validate age claims. We have studied methylation in all mammalian species, in hundreds of mammalian species. And we can estimate relative age, the age of an animal divided by the maximum lifespan of the species, using one clock, one pan-mammalian methylation clock. And we can study how environment, how stresses influence epigenetic aging. But we can also study how genetics influences epigenetic aging. And we have found that one mutation that produces a dwarf mouse is associated with much slower epigenetic aging, starting from a very young age. We have used methylation to study maximum lifespan in different species. We studied maximum lifespan in 348 mammalian species and wanted to find out what's the mystery behind exceptional long lives, like a bowhead whale can live for 211 years. And after years of work, I've come up with only one treatment, and unfortunately it only benefits half the population. And this is become female. So we found a female longevity advantage in 17 different mammalian species. We observed it in skin samples from humans and in skin samples from the bowhead whale. Now you probably wonder, well, how can I become female? And in this context, I want to highlight our studies in rams. We have found that castration delays epigenetic aging in ear samples of rams but has unfortunately no effect on blood samples or other tissues. So if you want to have young ears, you know what to do. Next, I wanted to study relationship and their influence on aging. Many of you may have heard the claim that married men age more slowly than unmarried men. But when I looked at the literature, the results were quite ambiguous and weak at best. So I turned to an interesting species that exhibits a monogamous mating behavior. Prairie voles remain attached to each other for years. And we compared the epigenetic ages of prairie voles that had successfully mated to prairie voles that were lonely and never attached to another prairie vole. And we looked at hundreds of samples in 4 different organs, and we found 0 effect. So to me, this is a hopeful message, because if you are lonely and depressed and unattached, at least you don't have to worry about your epigenetic clocks, you know. So, and it's better for you to be in a miserable relationship but follow a healthy lifestyle than to be in a happy relationship and follow an unhealthy lifestyle. So much I can tell you. However, are there special attachments that can rejuvenate you? And the answer is yes. Special attachments to younger mates. So if you attach the blood circulation of an old mouse to that of a young mouse, if you sew them together, several organs of the older mouse will be rejuvenated according to epigenetic clocks. Now, this is a joke. This is not a viable treatment for humans. You may have heard of ice baths, the new thinking, if you cool yourself, you slow aging. So I looked at this phenomenon in the context of hibernation and torpor. Torpor is a state where your core body temperature gets lowered and energy consumption will be lowered. And sure enough, when we studied yellow-bellied marmots, periods of hibernation were associated with slower epigenetic age. Sometimes you see torpor and hibernation depicted in science fiction movies in order for astronauts to overcome long distances of travel. Now, there is a way to lower body temperature in mice by stimulating special types of neurons. And we found that the long-term induction of this torpor-like state resulted in a 3-month age reduction in blood in these mice, which doesn't sound like much, but for a mouse, that is quite a reduction in epigenetic age. But what can you do right now? So we analyzed the blood samples from older individuals who had participated in an exciting clinical trial. So these people were 71 years or older, average age of 75, they were healthy, and we compared omega-3 intake, vitamin D intake, and a simple home exercise program. And sure enough, if you took participants who took 1 gram of omega-3 per day, they exhibited reduced epigenetic age, more precisely reduced GrimAge. GrimAge is our best mortality risk predictor, as indicated by the name Grim, as in Grim Reaper. But I was disappointed that the home exercise program did not result in epigenetic age reduction, because we all know exercise is healthy for you. And I thought, well, maybe these people didn't exercise vigorously enough. So therefore, I went to the other end of the spectrum. I analyzed Olympic champions. And interestingly, people who had won a medal at an Olympic event less than 10 years ago, their blood showed reduced GrimAge. However, champions who had won the medal over 10 years ago did not differ from control individuals. So that illustrates a point about the nature of epigenetic clocks. Unfortunately, rejuvenating interventions often lead to transient effects. There's a rebounding effect. And conversely, stressful events can accelerate your epigenetic age, but then after some recovery, it goes back to a normal state. So I hope I convinced you that epigenetic clocks allow us to characterize and study aging. We are using epigenetic clocks for identifying and validating different treatments that aim to slow biologic aging. I want to tell you, hang in there. There are a lot of exciting developments. I'm quite confident that sooner or later we will have safe interventions that slow your biological clock. Thank you very much.
Steve, now I know what to do if I want my ears to look young. Not quite sure if I'm ready for that stage yet. But beyond lifespan extension, what specific kind of age-related conditions do you think, do you believe might first benefit from epigenetic rejuvenation therapies?
That's a good question. So when it comes to epigenetic rejuvenation therapies, people often believe in these Yamanaka factors. There are certain transcription factors that rejuvenate tissues. And the concern is safety. So the key is, how do we prevent malignant transformation? And so people are looking, for example, at regenerating the optic nerve, because you can administer such gene therapies to the eye without the risk of it, these AAVs to infect other organs. Okay. So you need to think about how to make it safe.
Okay. You advise the audience to hang in there. I think everyone's down with that. Just want to kind of get a sense from you on the timeline you envision for translating the kind of the fundamental research you're doing at Altos Labs into kind of clinical applications for human rejuvenation. Like, how long do we have to wait?
It's so difficult, because sometimes developments are much faster than you expect.
Yeah.
And sometimes there are these breakthroughs that come from nowhere and much faster. For example, the AI revolution really surprised me, because I thought this sped us up by 100 years, To be honest, I have no idea, I'm hopeful. I believe that it can be done. That's why I work very hard on finding these interventions. I think it's worth my while to invest my time in it, but hard to predict. Yeah.
Well, thank you for doing the work you do, and we look forward to hearing some major breakthrough in the coming years.
Yeah, thank you so much.
Appreciate it. Thank you.